Hello everyone, welcome back to the session. So in the last class we have discussed about carbohydrates and we have completed and all the basic information has been covered under the carbohydrates. In today's class we are going to discuss about lipids. What is mean by lipids, what will be the types of lipids and what will be the structure and function of each and every lipids and all the important point of view with respect to our CUET exam. So first and foremost thing we have to know what is meant by lipids. So as of now just know that carbohydrates, proteins, lipids and all these things were coming under biomolecules. Without chemistry no biology will persist, right? See, lipids are heterogeneous group of hydrophobic or amphipathic organic molecules. So whenever we are discussing about the plasma membrane, we have discussed about the word hydrophobic as well as the amphipathic. See, so this is what a lipid bilayer. So the structure of plasma membrane is lipid bilayer, and one lipid, if we zoom out one lipid, this is phospholipid, and this we are having an hydrophobic head, so hydrophilic head and the hydrophobic tail. As both hydrophobic and hydrophilic will be present in the same compound, we are calling that particular the lipid as the amphipathic organic molecule. So this is what amphipathic. So like in the same way, this lipid is a heterogeneous group, which means a group of many different compounds together make up a lipid. So that is why a lipid is a heterogeneous compound of hydrophobic or it will be amphipathic organic molecules, but it is not hydrophilic exclusively. Now it is insoluble in water. This is very important. Just imagine a tumbler of water, and if you drop one drop of oil, and when the oil is not soluble in water, but instead it will be float on the water. But just imagine this is the tumbler of water, and you pour the sugar, and the sugar will be dissolved and it will be solved in the water, and water is the solvent. And this is what the difference between the lipid and the other compounds. The lipid is insoluble in water, but if the lipid is insoluble in water, do the lipids will soluble in any other solvents? Of course it will be soluble in some other solvents, and that solvents is called as the non-polar solvents. And what are all those non-polar solvents? That is nothing but an ether, chloroform and benzene. So these are all the this is how we can define what is meant by lipid. Now, what are all the general characteristic features of this lipids? So, what do we have studied when we discuss about the carbohydrates? So, it is composed mainly of carbon, hydrogen and oxygen, isn't it? The general formula of carbon dioxide will be CH2O of N. So this will be the general formula for the carbohydrate. So like in the same way, the lipid is also composed mostly of carbon, hydrogen and oxygen. But sometimes this phosphorus, nitrogen, sulfur will also accumulate with it. So this is what the lipids composed exclusively of this carbon, hydrogen and oxygen majority of the time, so yields more energy. So this is what the most important point with respect to this lipids. When compared to the carbohydrates and the lipids, carbohydrate has an instant source of energy, but the lipid will provide a more energy than the carbohydrate. So this is very important. For 1 gram we can get 9 kilocalories per gram. So this is very important. So when compared to carbohydrates, lipid gives us more energy, and that has to be in a good lipid. So stored in anhydrous form. So it is not a hydrous form. So it is stored in the anhydrous form. We are going to see what in which way they are going to store their material. See, not true polymers. We can't say this lipid are truly polymers. We are going to see what will be the structure of each and every lipid. And many lipids are esters of fatty acids. Many lipids are esters of fatty acids. So all these things were the general characteristic features of the lipids. So in the examination, how can we expect? So lipids are not polymers. Lipids are not polymers, frequently asked whether it will be a true or false statement. So lipid are not true polymers. Just note the statement because this is important. This can be expected in the exam. And see, This is what a plasma membrane. We know that. See, the plasma membrane, we have studied what will be the structure of the plasma membrane, and it is made up of a lipid bilayer, and two lipid layers together make up the plasma membrane, and that lipid is mostly, it will be phospholipid. Again, it is coming under the lipid. So the lipid is very important to maintain the structure of a membrane. So this is the first and the most important uses of this lipids. And also see, so in the first class we have studied that, yes, lipid, phospholipids. So in the phospholipids, between these phospholipids, this yellow color is nothing but a cholesterol. So this cholesterol is the one making the lipid membrane more rigid. So this is very important. So this cholesterol is also coming under the lipid, and the phospholipid is also a type of lipid. So the lipids plays an important role in maintaining the membrane structure. So this is the first uses of this lipid. Now, so this will be the types of lipids. Just to look at this flow chart, lipid can be classified into three major groups. One is simple lipid, compound lipid, and derived lipid. So this will be the three major types of the lipid, and this simple lipid can be further categorized into two, that is triglycerides and the waxes. So the triglycerides and the waxes will be coming under the simple lipid. And compound lipid, so we are going to discuss why it is called why we are calling this particular lipid as a simple lipid and this as a compound. We are going to see about that, but just know here how the lipids are getting classified. So see, the compound lipids has been classified into two, that is phospholipid and the glycolipid. Further, the derived lipid has been further categorized into three types, that is steroids, sterols, and carotenoids. So what will be the difference between the steroids and the sterols? Steroid is nothing but a lipid hormones, and the sterols, they are nothing but, there are different types of sterol. One major type of sterol will be the cholesterol and ergosterol. So all these were the cholesterol. The sterol which is present in animal is called as a cholesterol. The sterol which is present in the fungi is the ergosterol. The sterol which is present in the plants is stigmasterol. So all these were the types of sterol. Now another important compound will be the carotenoids. Carotenoids, so the best example, carotenoids is nothing but a pigment, and the example for the carotenoid pigment will be the beta carotene and xanthophylls. All these were coming under the carotenoids. Now this phospholipid, the example for the phospholipid is lecithins and cephalins. Altogether we are these were the example for the phospholipid. And in the glycolipid, this glyco, which means carbohydrate is associated with the lipid, is called as a glycolipid, and this glycolipid further classified into cerebrocytes and gangliosides, which plays a major role in the brain. Now this triglycerides has been classified into two. One is glycerol and fatty acids. So just like in the carbohydrates, see, we have categorized carbohydrates into three. One is monosaccharides, oligosaccharides, and polysaccharides. So whenever we are hydrolyzing the polysaccharides, we can get up to 10 to 20 monosaccharides. So the simplest form of carbohydrate will be the monosaccharides. So like in the same way, the simplest form of lipid will be the glycerol and the fatty acids. Whenever we are breaking the triglycerides, we can get two important compounds. One is glycerol and the fatty acids. Whenever a group of amino acids together, we are calling a protein. So you should know this carbohydrates, proteins, lipids, all these were considered as a polymers, and this carbohydrate is made up of many monomers, and each monomer we are calling them as the monosaccharide. And protein is made up of many monomer, and that monomer is nothing but an amino acids. And the lipids and group of glycerol and fatty acids, we can make up the triglycerides. So this is very important. And further, in this fatty acids, based on the bond they are having, we can categorize them into two. One is saturated fatty acids and unsaturated fatty acids. In this, unsaturated fatty acids has been further categorized into two. One is monounsaturated fatty acids and polyunsaturated fatty acids. See, The example for monounsaturated fatty acids will be the oleic acid, and the example for polyunsaturated fatty acids will be the linoleic and linolenic acid. So this is very important in an exam point of view. So this will be the overall flow chart with respect to the lipids. Now come to the classification of lipids. Now one by one we are going to see detailedly. So the first lipid will be simple lipid. So as we have mentioned in the previous slide, the lipid has been categorized into three types. One is simple lipid, complex lipid and derived lipid. And the first lipid will be the simple lipid. And the simple lipid, esters of fatty acids plus alcohol. So those are all the lipids which are simply the esters of fatty acids plus it will be associated with an alcohol. So all those lipids has been coming under the simple lipid. Say for an example, triacylglycerols, which is nothing but a fat and wax. Triacylglycerol and fats. See, waxes, the lipids, which means those lipids which can be the fusion of fatty acid and alcohol. So this alcohol is nothing but a glycerol. So glycerol and fatty acids, we are calling them as a simple lipid. And the example, the two things which is coming under the simple lipid will be the fats and the waxes. Fats are again nothing but a triacylglycerols. Now, what will be the compound lipids? See, very important thing. This complex lipids, either it will be associated with the protein or it will be associated with the carbohydrate. So that is what we are saying that as a compound lipids. This lipid is either it will be associated with the protein or it will be associated with the carbohydrate. So if it is associated with the other biomolecules, we are calling them as a compound lipids or complex lipids. Just see here, contain an additional groups that may be either phosphorus, nitrogen or carbohydrates. So phospholipids, glycolipids and lipoproteins. So all these were associated with some other biomolecule. So we have categorized them under the compound lipids. Now derived lipids. So these are all the lipids which has been derived from either simple lipids or the compound lipids. See, derived lipids, we can say that as a fatty acids, steroids, terpenes, prostaglandins. All these were categorized under the lipids only, no doubt about that. But it has been derived from the simple or the compound lipid. It has been associated with the simple and the compound lipids. So that is why we are saying that as a derived lipids. So usually these questions has been asked in the match of following questions. Whether so they have given here as a simple lipid, compound lipid and the derived lipid, and here they give one of the examples. It can be expected in the match of following questions. Kindly make a note of that. Now, so we have said that fatty acid plus glycerol together we can make up the simple lipid. So we can so we should know what is exactly the fatty acids. So very important definition. Fatty acids are long chain monocarboxylic acid with hydrocarbon chains. Just look at this, fatty acids are long chain, again very important, and monocarboxylic acids with hydrocarbon chain. Hydrocarbon chains in the sense they contains only hydrogen and carbon. So that is what we are saying, hydrocarbon chains. So usually generally this fatty acids are long chain hydrocarbon chains. So this is what the perfect definition for the fatty acids. Now we have studied two different types of fatty acids. That one will be the saturated fatty acids and another one will be the unsaturated fatty acids. And the saturated fatty acids, we are having a single bond. We are having a single bond. In unsaturated fatty acids, we are having a double bond or we are having a triple bond. So these double or triple bond, they were coming under the unsaturated fatty acids, and with the fatty acids which contains a single bond, they are coming under the saturated fatty acids. And one of the most important feature of the saturated fatty acid will be they will be solid in the room temperature. Whenever the fatty acids are whenever the fatty acids which is present in the room temperature, these saturated fatty acids will be solid, and it has a higher melting point. The melting point will be higher, and the example for such fatty acids, such saturated fatty acids will be the palmitic acid and stearic acid. So this is very important. The 16 carbon containing compound, 16 carbon fatty acid will be the palmitic acid, and 18 carbon fatty acid will be the stearic acid, and both of these acid will be coming under the saturated fatty acids. And one of the most important point in the exam point of view is animal fats are saturated. Animal fats were saturated. Now comes to the unsaturated fatty acids. Just look at the picture. We are having an oleic acid, linoleic acid and linolenic acid. So all these were coming under the polyunsaturated fatty acids. See, just look at this oleic acid. This is the structure of this oleic acid. We are having a double bond here and we are having a double bond here. So these are all the acids it will be coming under the unsaturated fatty acids. It is not one double bond or one triple bond. It will be one or more double bond. One or more carbon to carbon double bond will be present in the unsaturated fatty acids. So what do we have studied with respect to the saturated fatty acid? It will be solid at room temperature, but in the unsaturated fatty acid it will be liquid at room temperature. Further, this unsaturated fatty has been classified into two. One is monounsaturated fatty acid and polyunsaturated fatty acid. And this the example for this monounsaturated fatty acid will be the oleic acid, isn't it? So the number of carbon atoms present in this oleic acid will be the 18 carbon. Now polyunsaturated polyunsaturated fatty acids, that is PUFA, linolenic acid and linolenic acid, arachidonic acid. This is the C20 20 carbon containing fatty acid will be the arachidonic acid. So these are all the very important examples of this unsaturated fatty acids. We keep on expect question from these things. And linoleic acid, the carbon atoms will be the 8 carbon atom will be 18, and at 6th place we are having the double bond. Now this is what a structure of fatty acids. See, fatty acid are made up from the carboxylic group and the hydrocarbon chain. No doubt about that. Carboxylic group in the sense COOH. So this is what the carboxylic group. So just look at this picture. This is what a fatty acid. So in this fatty acid head, that is in the hydrophilic region, it is made up of the carboxyl group. That is what COOH. Right, now just see here, this is what the hydrocarbon chain, where we have only carbon and hydrogen. So as it is made up of only carbon and hydrogen, we are saying this as a hydrocarbon chain. Now so just look at this picture. We are having two different types of fatty acid. One is saturated fatty acid and another one is unsaturated fatty acid. In saturated fatty acid, we are not having any double bond or triple bond in their structure in case of saturated fatty acid. And just here, unsaturated fatty acid, definitely there will be the double bond. The double bond will be present, but not at only one place. It may be one or more places. Now, essential fatty acids. So this essential fatty acids cannot be synthesized by humans. See, in amino acids, whenever we are going to study in the future, we are going to study about the amino acid and protein, and at that time we are also going to study about what is meant by essential amino acid and what is non-essential amino acids. Like in the same way, in the fatty acids also we have essential fatty acids, and those essential fatty acids cannot be synthesized by the humans. And that essential fatties will, the example for the essential fatty acids will be the linoleic and linolenic acids, and both of these acids will be coming under the unsaturated fatty acids. And we have studied this oleic acid, again it will be coming under the unsaturated fatty acids, but it is the monounsaturated fatty acid. And just see here, and in oleic acid is not essential. Oleic acid is not an essential fatty acid. This linoleic and linolenic, which is coming under the polyunsaturated fatty acid, which is very essential fatty acid. And just remember, this essential fatty acid cannot be synthesized by the human beings. See, this is what the structure as we know before. So this is what the head part. So this is what the head part, the hydrophilic head, and this is the hydrophobic tails. So in the hydrophobic tails, one tail region is the saturated fatty acid and this structure is the unsaturated fatty acid. Those in the unsaturated, this kink-like structure, the bend-like structure of this chain is because of the double bond which is present under the unsaturated fatty acids. See, we are having two tails and all those tails we are saying that as a hydrophobic tails. In the hydrophobic tails, and definitely that will be the fatty acid, and we are having two fatty acid, one a saturated fatty acid and unsaturated fatty acid, and the saturated fatty acid chain will be very straight and the unsaturated fatty acid is a little bit kink-like structure or bend structure. So why this bend structure is because of the double bond present in the fatty acid. So this is what this is the structure which maintains the fluidity. So wherever, just look at this point, why we have to maintain the fluidity and rigidity in the membrane. This is very, very important. See, so we have studied in the first class itself. So the lipid bilayer, if the temperature is higher, the lipid membrane, that is a plasma membrane, will be more fluid. If the temperature is higher, the membrane will be more fluid. If the temperature is very lower, which means in the colder region the temperature is very low and there the membrane is very rigid. And you should know the membrane should not be extreme fluid and the membrane should not be extreme rigid. And who is going to taking care of preventing this too much rigid and too much fluidity? And that will be done by the most important factor. We are going to discuss about that. See, that will be, and the temperature is higher in this region. As a result, the membrane fluidity is also more. And to maintain the rigidity, to reduce the fluidity, there is a presence of cholesterol. As I have mentioned, as I have shown in the previous slide, in the first few slides, there is an yellow color structure which is embedded between the saturated and unsaturated fatty acids. Just look at this picture. Just see here. So this is what cholesterol, which makes the membrane rigid. See, at if the temperature is higher, the membrane is more fluid, and to prevent the membrane fluidity, there is a presence of cholesterol, which makes the membrane rigid. And at the colder region, the temperature is very low, and as a result, as the temperature is very low, the membrane is also more rigid, and the membrane should not be more rigid, and there the fluidity has been enhanced by this unsaturated fatty acids. How the unsaturated fatty acids is making the membrane more fluid? Because of this kink-like structure. See, look at this picture. It will be very tight when it will be like this. When it will be like this, there is some gap present between this one lipid between to the another lipid. So this is what this kink-like structure of this unsaturated fatty acids makes the membrane more fluid. So this is very, very important in an exam point of view. Now, see here. So this is what the hydrophilic head is made up of phosphate plus glycerol. So glycerol, which is nothing but an alcohol, and this is a fatty acid. Alcohol plus fatty acid together we can call them as a lipid. So this is what an outside of the cell, and this is the inner side of the cell, and there is a protein which is embedded within it. And all these explanations regarding this plasma membrane have been discussed in the first class itself. Now coming back to the simple lipids, and the most important simple lipid will be the triglycerides. And the triglycerides, whenever we are breaking this triglycerides, we can get two important monomer. One is glycerol and 3 fatty acids. Glycerol, 3 fatty acids, that is why the name triglycerides. If there is a glycerol plus 2 fatty acids, it will be diglycerides. If there is a glycerol plus 1 fatty acid, that will be monoglycerides. So this is why, which means monoacyl glyceride, diacyl glyceride, and triacyl glyceride. So here the triacylglycerol, which is made up of glycerol plus 3 fatty acids, this is very important. And this glycerol, triacylglycerol, many lipid will be stored in the form of triacylglycerols. Kindly make a note of it. This can be expected in the question. Alright, see, the major storage lipid that will be the triacylglycerols, which is also called as the triglycerides, and it is stored in the adipose tissue. What will be the function of this triacylglyceride? It will be the energy reserve, and it will be the thermal insulation and protection of organs. So all these were the function performed by this triacylglycerides, and this will be the major storage lipid stored in the adipose tissue. See, this triacylglycerides are neutral lipids, so this can be expected in the question. Now, waxes. So, waxes is made up of long chain alcohol plus fatty acids. So, this can be expected in the exam. So, triacylglycerol is made up of glycerol plus 3 fatty acid, and whenever we are breaking the waxes, we can get the fatty acid and the long chain alcohol. It is not a short chain, it will be long chain alcohol, and it will be highly hydrophobic. It is highly hydrophobic. So as it is water hating, we can call this waxes are waterproofing. So the best example of this waxes will be bee wax, cutin, which is present in the plants, and ear wax, that is cerumen. So these were the waxes. Very important thing. Now that's all about the simple lipids. Now comes to the compound lipids. So the compound lipids, it is the example for the compound lipids will be the phospholipids. Okay, so this phospholipids, it has 2 fatty acids. Just now I have shown the picture. So this is what the head of the phospholipid, and this is where the tail of the phospholipid, and the head region will be called as the polar head group, which means hydrophilic, and the tail group is the hydrophobic fatty acids, and in one head we are having 2 tails, so that is why 2 fatty acid, one saturated fatty acid, one unsaturated fatty acid. That's why 2 fatty acids, and in the head region we are having an alcohol and the phosphate, so that is why we are saying glycerol backbone, and there is a phosphate group, polar head group. Right? So as they are having, see, just look at this picture. It has a hydrophilic head, and they are having a hydrophobic tail. As both hydrophobic and hydrophilic present in the same compound, we are calling this phospholipid as the amphipathic in nature. So this phospholipid is majorly involving in forming the membrane of the plasma membrane. Now, different types of phospholipids. Different types of phospholipids are there, and the first type will be the glycerophospholipids. And the glycerophospholipids, again it will be an umbrella term. Again there will be different types of glycerophospholipids, that is lecithin, cephalin, phosphatidyl-serine, phosphatidyl-inositol, and cardiolipin. So if the glycerophospholipids, if it is made up of phosphatidylcholine group, we are calling them as a lecithin. And in the cephalin is again the type of the glycerophospholipids, it is called as the phosphatidyl-ethanolamine, and phosphatidyl-serine and inositol. And this cardiolipin we have studied when we discuss about the cell organelle mitochondria. We have studied the inner mitochondrial membrane is made up of this type of lipid, that is cardiolipin. And this cardiolipin, this is very important in an exam point of view. Kindly make a note of it. Now, if the phospholipid present in the nerves, that is the myelin sheath, that phospholipid is called as a sphingophospholipid. So the sphingophospholipid, the type will be the sphingomyelin, and sphingomyelin is very important for the formation of the myelin sheath. This is also again important. So just see here, this is what the structure of the sphingosine, and see here it is ceramine. See, this is exactly look like the sphingosine. So sphingosine backbone, and again there will be an addition of fatty acid residue. So the sphingosine plus a fatty acid residue, we are calling them as a ceramide. This is very important. Just look at this picture. This is exactly this picture. This is what the structure of sphingosine. And together with the sphingosine, if the fatty acid residue has been added with the sphingosine backbone, we are calling that particular structure as a ceramide. And a sphingomyelin, again sphingosine backbone with this fatty acid residue. If it is associated with the phosphocholine group, we are calling them as the sphingomyelin. Just look at this picture. We are having a sphingosine backbone and we are having a fatty acid residue, that is a ceramide. See, sphingosine, this is the structure of the sphingosine, and sphingosine plus a fatty acid residue, we are calling that as a ceramide. Now there is a sphingosine backbone and the fatty acid residue, together we are calling them as a ceramide. If the ceramide is further associated with the phosphocholine group, we are calling that particular term as a sphingomyelin. And in the ceramide plus a single sugar residue, we are calling them as a cerebroside. So all these were the structure of different types of phospholipids. Now this is what a ganglioside. So in the ganglioside, there is a sphingosine. See, there is a sphingosine, and here we are having a fatty acid residue, and it is not a single sugar residue, it is oligosaccharide residue, so which means a cerebroside. So this is what we are calling them as a cerebroside, and just a modification of a cerebroside, a sphingosine backbone and a fatty acid residue here, and an oligosaccharide residue plus sialic acid. Altogether we are calling them as a ganglioside. These are all the most important phospholipids. Now comes to the glycolipids. So if the word glyco refers to carbohydrate. If the lipid associated with the carbohydrate, we are calling them as the glycolipids. Here there will be no involvement of the phosphate group, and this glycolipid will be seen majorly in the nervous tissue. Right, so this is important, and the type of glycolipids were the cerebrosides and gangliosides. Just now we have seen the structure, a sphingosine residue plus a fatty acid plus single sugar residue, we are calling them as a cerebroside. And a sphingosine backbone plus fatty acid plus oligosaccharide residue plus the structure plus sialic acid. Together we are calling them as the ganglioside. So the sialic acid will be present in the gangliosides. So the function of the glycolipids will be the cell recognition and nerve conduction. So this will be the major function performed by these glycolipids, and this can be expected in our exam. That is, gangliosides contain the sialic acid. This is very, very important. Now just look at this. Transport lipids in the blood. So who transports lipids in the blood? That is nothing but a lipoproteins. The lipid which is associated with the protein is called as a lipoproteins. Now those are all the lipoproteins are chylomicrons, VLDL, LDL and HDL. So this chylomicrons, here a major lipid will be the triacylglycerol. Now what will be the function of this chylomicron which is made up of this triacylglyceride? They are the one transporting the lipid. After the digestion, the lipid will be the lipid is also getting digested. The digested lipid will not be absorbed by the blood because the blood cannot be absorbed the lipid, but instead it will be involving the chylomicrons. They are the one functioning the lipid transport. So this is very important. VLDL, very low density lipoprotein. That is what VL. Once again, VLDL stands for very low density lipoprotein. Again, this VLDL is also made up of a triacylglycerol. So in the triacylglycerol, their function will be from the liver to the tissues, it is getting supplying. Now LDL, low density lipoprotein. Low density lipoprotein, this is the one, the major lipid here, it will be the cholesterol. So the cholesterol is said to be as a bad cholesterol. So this is very important. Now just see here, HDL, high density lipoprotein. So this high density lipoprotein, majorly it contains a protein. And all these protein associated with the lipid, we are calling them as a good cholesterol. So this is very important table. This can be expected in the exam. Now, the derived lipids. So, what do we have discussed about the derived lipids? Those are the lipids which is derived from the simple or the complex lipid. We are calling them as a derived lipid. And in the derived lipid, we have studied about the steroids, sterols and all those. Steroids are having 4 fused rings. The structure of steroids will be 4 fused rings, and there will be no fatty acids will be present in the steroids. Just look at, very important. No fatty acids will be present in the steroids, and in the lipoproteins, sorry, the glycolipids, there will be no phosphate. No phosphate. So generally in the phospholipid there will be the phosphate, there will be an alcohol, there will be a 2 fatty acid chains. So this is how a phospholipid will be, and here it is a glycolipid which has no phosphate group, and in the steroids they are having no fatty acids. And what are all the important steroids? That will be cholesterol, bile acids, steroid hormones, vitamin D. All these were an important steroids. This is very important. And cholesterol, which is coming under the derived lipid, and this cholesterol which is a component of the cell membrane. This is very important. Just now I have mentioned you the extreme fluidity of the membrane has been controlled by the cholesterol. So the cholesterol is enriching the membrane's rigidity, and this cholesterol is a precursor to form the bile salts and the steroid hormones. So cholesterol is also important only if it is good cholesterol. So cholesterol is a precursor of the bile salts and the steroid hormones. The cholesterol stabilizes the membrane fluidity. So if the membrane is more fluid, the cholesterol is making the membrane stable by making the membrane more rigid. So this is why we are saying the cholesterol stabilizes the membrane fluidity. Now, so this is what the structure of cholesterol. Just now we have studied in the steroids we are having 4 fused rings. 1, 2, 3, 4. So we are having 4 fused rings. Again the cholesterol is coming under the steroids. Now eicosanoids. So they are derived from the arachidonic acid. Very, very important. This can be expected in the exam. So eicosanoids, again it is coming under the derived lipids. It is derived from which acid? Arachidonic acid. So this arachidonic acid, where do we have studied this? This is coming under the unsaturated fatty acids, which is the simple lipid. So this eicosanoids will be derived from the simple lipid. And what are all the types of eicosanoids? That will be prostaglandins, thromboxanes and leukotrienes. All these were involving in the, so this prostaglandins will be involving in the inflammation. Thromboxanes, it will be involved in the pain reliever, and the leukotrienes will be involving in the platelet aggregation. So all these were, prostaglandins will be involving in inflammation, leukotrienes it will be the pain, and thromboxanes will be the platelet aggregation. So all these were the important functions of each eicosanoid. So this is very important, and this eicosanoids has been derived from the arachidonic acid, and we have studied this under the fatty acids, which is the simple lipid. Now, what are all the biological functions of these lipids? So, lipids are majorly for energy storage. Say for an example, the excess glucose will be converting into a glycogen, and this glycogen is nothing but a lipid. Structural components, this glucose, glycogen, and that's completely irrelevant to this. And here the energy storage is the main function of this lipids. Structural component of the membranes, insulation and protection. So insulation and protection, hormone synthesis. Many lipid based hormones are present in our human body. We can call them as a steroid hormone. So for the hormone synthesis, lipids plays an important role, and for the signal transduction, it is very important. And in the vitamins, we are having 2 types of vitamins. One is water soluble vitamin and another one is fat soluble vitamin. And the fat soluble vitamins are A, D, E, K. So all these fat soluble vitamins has been absorbed by these lipids. So all these were the functions of the lipids. So this can be expected in the exam. So A, D, E, K will be coming under the lipid soluble category, lipid soluble vitamin, and B and C will be coming under the water soluble vitamins. Now, how the lipids is getting digested and how it will be absorbed. The lipid has been digested by the bile salts. bile salts are involving in emulsification of fats, which means fat digestion. That's all. Simply just remember here, the bile salts are involving in the emulsification of fats. Emulsification in the sense digestion. And the pancreatic lipase, in the pancreas we are having an enzyme which is exclusively made for the synthesis digestion of the lipids, and that is nothing but a lipase. It is present in the pancreas, so that is why the name pancreatic lipase. So this is the enzyme which is breaking the triacylglycerides into fatty acid plus monoglyceride. So this will be the simplest form of the lipid, and it is absorbed as micelles, and that micelles is nothing but a kilomicron. So re-esterified into kilomicron. So the transport material or absorbing material of the lipid will be the kilomicrons. And one more important point, the bile salts is involving in emulsification of fats, that is the fat digestion, so that you should not think that this bile has some enzymes. No, the bile has no digestive enzymes. So this is very important here. Now, another important thing will be what will be the difference between the white fat and the brown fat. So, the white fat is involving in energy storage, and the brown fat are involving in the production of heat. And the mitochondria in the white fat are very few, but in the brown fat there will be more mitochondria within it. And UCP1 is absent in the white fat and it will be present in the brown fat. So as the brown fat is involving in the heat production, so the technical term for the heat production will be the thermogenesis, which is nothing but a brown fat done by this brown fat. So these were all the important one-liners with respect to the lipids, as lipids are not polymers. Essential fatty acids are polyunsaturated fatty acids, but not oleic acid. Oleic acid is a monounsaturated fatty acid. So this point is very important point. And phospholipids are amphipathic, as both hydrophobic and hydrophilic will be present in it. So cholesterol is coming under the derived lipid, as it is coming under the sterols. Cardiolipid, which is present in the inner mitochondrial membrane, and HDL is a good cholesterol, and HDL is a good cholesterol, and LDL is a bad cholesterol. Just know this. So this is where the question can be trapped as in the question paper. The lipids are soluble in water. No, it is insoluble in water. Oleic acid is essential fatty acid. Just now we have studied essential fatty acids are polyunsaturated fatty acid, and this oleic acid is coming under the monounsaturated fatty acids. Waxes are storage lipids. No, waxes are not storage lipids. Bile is an enzyme. Bile is not an enzyme. It is a secretion secreted by the liver. And cholesterol is triglyceride. No, cholesterol is a sterol. Now, so that's all about the most important things with respect to this lipid. Now comes to the question. So which of the following statements about the lipid is correct? Lipids are the polymers of fatty acids, insoluble in organic solvents. Lipid provide more energy per gram than carbohydrates. Lipids are hydrophilic in nature. Let us rewrite all these statements. Lipids are the polymers of fatty acids. No, lipids are the polymers of fatty acids plus glycerol. And lipids are insoluble in organic solvents. No, it is soluble in organic solvents and insoluble in water. And it provides more energy per gram than carbohydrates. Of course, this is the right statement. And they are hydrophilic in nature. It is both hydrophobic or amphipathic, but not hydrophilic. Now, the major structural lipid present in the inner mitochondrial membrane is none other than the cardiolipid. Just now we have discussed about that. And which of the following fatty acids is an essential fatty acids in case of human, and that will be the linoleic acid. An amphipathic nature of this phospholipid is due to the presence of, why it is amphipathic? Because both hydrophilic and hydrophobic is present here, as that is a polar head group and non-polar tail group is present. It is amphipathic. And which lipid is mainly responsible for insulation of nerve fibers? So insulation of nerve fibers. So where exactly the nerve fibers is made up of? And the answer will be sphingomyelin. So sphingomyelin is an important lipid which is responsible for myelin sheath formation. And here, which of the following lipoproteins transport dietary triglycerides from intestine to tissues? That will be the chylomicrons. The cholesterol differs from the triglyceride because cholesterol contains the fatty acids, is an ester, has a 4-ring steroid nucleus, is a storage lipid, and the answer will be option C, no doubt in that. Brown adipose tissue differs from white adipose tissue by the presence of higher lipid droplets, fewer mitochondria, uncoupling protein UCP. 1, absence of vascular supply, and the answer will be uncoupling UCP1. Which of the following is not a function of lipids? It is a long-term energy storage, structural component of the membrane, genetic information storage, and hormone synthesis. Genetic information storage is not done by the lipids. And bile salts plays an important role in lipid digestion. Bile. Hydrolyzing the triglycerides? No. Acting a lipase? No, because it doesn't have any of the lipid digesting enzymes. Absorbing fatty acids? No, it is involving in the emulsification of fats. Okay, so that's all about this today question. So in today's class we are completely discussed about the lipids. We'll meet you in the tomorrow's class. Thank you so much.